Okay, we're going to keep rolling right along here, and not to forget about the other sectors that we have here on Prince Edward Island, which I tend to do when I focus basically on potatoes. And Joanne Driscoll from the PEI Horticulture Association tries to keep me in line. Joanne's been involved in a number of studies and research projects, and Joanne's going to present some of her data on a multi-year trial that she did, her and her team did here on Prince Edward Island. So I'd like to welcome Joanne to the podium to make the presentation. Want to use this, or you want to?
Yeah. Thank you, Brian. Hello, everyone. If you're like me, you're pretty well super saturated about this point in time. It's sort of hard to stay a little focused. My staff, though, have a cue. I shouldn't say my staff, my coworkers. They know what to tell me when I get off focus. Okay. So basically, as far as the Hort Association, and we are the rest of the crops, I started back in 1986. And basically, we did see wireworms back then. It was very isolated. We knew if we went into a sod field, that's exactly what we were going to find. Then throughout the early 2000s and leading into 2010, we kept seeing a massive increase in losses. It was scattered throughout different fields, different crops. So we went back in the end of 2010 and we looked at our historical data that we had been collecting since Christ was a cowboy. And basically, we were looking at our crop scouting data, that sort of thing, and looking at the trends. So we did see, and we showed where there was an increase in damage. We also saw that we were seeding later, our carrots in particular. We had also decreased the use of our earlier pesticides, whether it be for carrot rust fly or weevil or what have you. Also, our products, the line that we had at our disposal had decreased. So everything that everyone has said today is the exact same in all the crops: carrots, onions, cabbage. So from there, we decided, though, that we needed to develop a study, and along with Bob's thinking, we want to look at the two modes of attack, battle from the two fronts, from both the click beetle as well as the wireworm. Ooh, I forget I have to do two things here. You know I'm a bit scattered, so bear with me. This was a big project. Half the data, we're still trying to look at it and figure out how we can relate it to our crops and our growing system here. So it was broken into three parts. We have the click beetle. We did the efficacy trials with the great assistance of Dr. Christine Arona. Thank you very much, Christine. She covered that earlier. Then next, we have our adult monitoring, and we increased that investigation in 2013. The second part was our crop rotation. We looked at the use of brown mustard and buckwheat as compared to our conventional crops that we were using for rotation. We used Christine's model for the protocol, but also what we did is instead of doing 2 years only of both those rotation crops, we included the 1 year rotation, and that was in the final year before our commercial crop. The conventional crops we used were barley, underseeded clover, we had timothy, and we had wheat followed by rye. The third part of our study was crop response. There are no tools for us as far as insecticides in my carrot world and rutabaga and cabbage. So basically, we do have to look at crop rotation in our world as far as the larva stage. So in some of the literature reviews, what we had done, they were giving us contradictory information as far as what would happen to our yields. So a very key part to us was, if we're using a 1 year or 2 year rotation, how is this going to affect our crop yield in the end? As far as the click beetle, this was a major part, and it's always been near and dear to my heart. The collecting of beetles in 2011 and 12, we did them primarily for the efficacy trials that Christine was doing in the lab. On 2013, we added a weather station, Watchdog 2000 series, and it went to our one site. This was our big site. It had a heavy infestation in 2009. We had also set up our buckwheat brown mustard plots there. It was a 10-acre field, and we put 7 traps along the perimeter of the field. We used sputatter specific lures, and we had the weather station going. The weather station was monitoring air temperature, precipitation, also soil temperature and soil moisture at 3 different depths, at 10, 15, and 25 centimeters. So the massive data we have there is just insane, and we pray Christine will help us work through that. So, however, what we did was we set the traps out on April 22nd. The weather station was on April 29th. This here, and it's, sorry, I'm looking at a screen versus a big screen, and anyway, we have our click beetle trapping, and this is what we found. So we put our first trap out the end of April. May 1st was our very first catch, and we had 10. She peaked on June 17th, and you can see she went up to 570 adults. And remember, this is sputatter specific, males only. And we monitored all the way to October. But our last catch was on August 20th, and that was a catch of 8. Another thing I'd like to note is that the mean soil temperature when we first started was 9 degrees Celsius. And we had an average range of air temperature between 8 and 22 centimeters throughout the whole span of their emergence. We feel there is more work warranted here, and I'll get into the more specifics of what we'd like to do next year. But we really do, by seeing these catches, and in particular in 2012, when we were monitoring and collecting, the spike, or I shouldn't say spike, we found a lot higher counts earlier on in 2012. On May 8th in 2012, we found 247 beetles as compared to your 58. So they're showing quite a difference there as far as when we might be seeing some peaks and that sort of thing for actual field spraying. So wireworms and cabbage. You've seen a few of these pictures before with Christine's presentation. The cabbage was a crop that was chosen, and it's due to the extreme high losses that we are experiencing in the spring with our transplants. This here picture is from one of the hot zones in the survey. The wireworm is killing the plants. It's going after the young rootlets when we put them out. The plant is not sturdy enough to combat the intensity of a high population. Sort of like the cabbage root maggot that we experience as well. When the plant is young, it cannot put out enough rootlets to survive, I guess is the word I'm looking for. This here is also compounded in our early transplants with the effect that we have crows coming in, and they are going after the worms, so they're pulling the plants out to get at the worms that are in the ground. That's when you experience losses such as this, where we have heavy misses, loss of plants. What this does is it's requiring more labor and cost to actually go in and replant the field. The second thing that happens is we have an uneven plant stand. So if we go in and put our plants back in, we still have an uneven growth. Some plants don't mature, so we've decreased our yield. We've increased the number of passes that we have to make across the field. And remember, we hand harvest. So thus, your cost of production has been increased. So that's why the priority was put on this. So this is the spring feeders. Briefly, I'm going to run through all these slides. What we have is, this here is showing the number of plants with wireworms. What we did was, weekly they went in after transplanting, and they, I mean, the people who did all the work, which is Andrew and Shelley. So they went in every week. They pulled 10 plugs randomly per each replicate. This here site had 4 replicates, 3 sub-treatments. But anyway, they went in, 5 treatments. They went in every week, 10 plants, pulled the plugs, counted the number of plants with plugs. So as you can see, the first date we caught was May 22nd. The soil temperature was at 11 degrees there at that point in time. We sampled all the way until June 10th. You can see that also after May 29th, all the counts dropped. Okay. We do have the highest count was in our, sorry, I'm getting so confused with these numbers, but basically what we have is the best control came with the 2 years buckwheat. You can see on the far side, we have 8.8%. The conventional was midway. 16.9% of your plants had wireworms in them. Whereas you have your 1 years coming in and your 2-year brown mustard coming in midway. Did that make sense? No, I'm not saying it right, but anyway. You can see that what we came out with was the 2 years buckwheat did the best in this scenario. Okay, then we move on to wireworm damage in carrots and rutabaga. Carrots was 2 sites. The rutabaga was 1 site. They did field assessments for damage twice per week starting in late August. Then we did a fall harvest. The carrots were harvested on September 30th and the rutabaga were on November 6th. They graded the plot in 2 ways. 1 sample was taken. They were graded for marketability. We ignored the wireworm damage, and that was to get our crop response. The next way, then all those were taken and divided into wireworm damage and no wireworm damage. Okay, so we looked at it in 2 ways. Basically, with your rutabaga damage, this is the percent roots damaged, and this is a mean of all the treatments. Your best results was the 2 years brown mustard at 26%. And then that was followed by your 1 year brown mustard at 29. Your buckwheats were midway with the 33 to 39%. And your conventional crop had the highest damage at 47.8%. Damage. Okay. And we will come to a trend here in a second. Carrot damage. Same type of thing. We had 2 sites, 2 replicates per site, 4 replicates per treatment, 4 subplots within each replicate. We did damage both by weight and by number. The best results in this scenario was your 1 year brown mustard, and then followed closely by the 1 year buckwheat. When I say 1 year and 2 year, both the 1 year and the 2 year rotations had 2 plantings per year. We followed Christine's protocol. The conventional crop, once again, resulted in the highest damage at 10.4 and 13.3. When we look at crop response, we contradicted most of the research out there, and our yields were all the same. Didn't matter which crop, whether it was 1 year, 2 year, 3 years, there was no statistical difference between these treatments. The carrots resulted in a range of 52 to 58 tons per hectare, early cabbage 23 to 40 tons per hectare, and the rutabaga was 11.8 to 20.1 tons per hectare. So our conclusion came to, for this spring feeding, the early transplanted cabbage, the 2 year buckwheat gave us the highest control. In the rutabaga, for fall feeding, the 2 year brown mustard worked the best. In the carrot, it was the 1 year brown mustard. And overall, in both the carrot and rutabaga, the conventional crop rotation resulted in higher damage. So we had higher damage, same yield, but more damage. So in all cases for the fall, our brown mustard and buckwheat worked better. For next steps, we would like to see efficacy trials continue on the beetle. We also feel that we should be doing some work on some degree day and possibly on the movement, where Bob was saying they're in the field, out the field. Let's confirm a bunch of this and see what is happening so we can come up with some really good strategies. We'd also look at, we have all sorts of ideas going on in our office. One of them would be for crops that would attract or repel. So whether we want to pull all those beetles or we want to repel all those beetles. So these are sort of theories we're working on. We would also like to, we have GPS'd all our sites and all our baiting. We did baiting and lures and et cetera. We'd like to go back into those if those fields are available, because they were actually rented fields that had big problems in 2009 and 2010. So the farmers rented the fields for us. But if they are available, we'd love to go back, maybe put some more baits out, see what's going on, see did we affect that population long term in the field. And as everyone has said, it is a team effort, and we could not have done this big project. I only presented a few little aspects of what we gathered. We had Dr. Nerona, Christine, you put up with us for 3 years. Thank you very much. She kept us focused. Andrew and Shelley did all the work. We had Dave and Donnie put up with us constantly on the cell phone. Our growers were so patient with us, and the inputs and the value of everything they put in, from the crops to the fertilizer. And I will stress Christine's point, the brown mustard and buckwheat do need to be fertilized. We saw a big difference when we used the fertilizer. Also, we were funded through this project under the ADAPT Council, came in year 3. The first 2 years were under GF1 with the PEI Department of Agriculture's Agricultural Research Fund. We also had Scott Anderson, Brian Murray, Dewey Pratt, and the crew. I feel like I'm at the Oscars. And Kevin Sanderson. But like, seriously, all of these people came together. Shauna, Brian, everyone helped us. And we hope that you'll continue to work with us on this. But it really was a big endeavor for us. I think we found a lot of information out. This information session today actually reaffirmed a lot of stuff for me. Also got my brain moving, which is where I'm having trouble concentrating, because now I'm thinking of even more ideas that I'd like to try. I understand Ted's enthusiasm and his passion, and I agree with when Bob's talking about understanding the behavior and coming with the 2 approaches of both the click and the wireworm. And I really believe in Christine and all your work and how you can keep us focused, and hopefully we can come up with a strategy. Thank you, and it's the end of my result.
Any questions for Joanne?